Hello everyone. I hope I'm audible. So before I start, let me introduce myself. I'm Vani Kari, biology educator at Unacademy. I'm an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. So let's get started. So just a second. Yep. So join with us in Telegram for homework and class notes. You can join the channel Vani Kari. One second, yeah. So Vani Kari class 9 and 10 biology, and the Telegram channel of Unacademy, ICSE and ISC both. So this link you can find in the description box. So you can click on the link and join the channel for updates regarding the classes. So special classes, so these are free classes conducted on Unacademy platform. So you will have access to all of this. You need to use the code. If it asks a code to unlock, so you can use the code Vani YT. So this is the code. So with this code, you can unlock many of the free products that are there on Unacademy. Alright. So learn from your favorite educator for free because these are free classes. But here in free classes, only a certain percentage of syllabus will be covered. So if you are planning for the a whole class 9 or class 10 completion, or it can be with respect to ISC also, so you can go for the plus subscription. So engage with educators via poll. Clear your doubt in the live special class. So basically we'll be conducting a quiz. So at the end of the quiz, so we'll be sharing a leaderboard so that you can know your rank and compete with your friends. Clear your doubts in the live special class. So for this, basically there is a question tab separately, so you can type in your question. You have an option to attach an image and send it to the educator during a live session, so they'll be getting a notification. So for the top special classes, you can visit the link in the description. So now Unacademy subscription features, so you can learn live from the comfort of your home. Unlimited access to all the courses will be there. Different educators will come up with different courses, so you will have access to all the courses once you take the subscription. Then top educators of India are going to take the classes in one platform, that is Unacademy platform. There is regular doubt clearing session, after about 3 session, the 4th session will be your doubt clearing session. So whatever doubts you have related to the topics that are previously covered in this session, so you can clear your doubt in this session. So that is a doubt clearing session. Also it could be a answer writing session if you do not have any doubt, or let's say all your doubts are cleared, so we can conduct some quiz, or maybe a discussion session can be done. Then complete syllabus will be covered. Then mentorship and guidance will be given. Study material is given. The practice tests are given. Live test series, and along with that you will have access to batches. Now we have batches in English, batches in Hindi. Okay, so that is English basically. So you will have access to batches, all the batches once you take the subscription. So but depending on your comfort, you can go for any of these batches. Okay, so there is, along with this, again there is daily practice section, weekly mock test series, live quizzes will be there, daily MCQ and subjective test series will also be there. Okay, so now you can have a look at this. So since this is your very beginning of your class 10, so you can go with, so this is for class 10, so you can go with 12 months of subscription. If you just want to try how exactly this works, so for 3 months also you can go, or maybe 6 months. So this is completely your choice. So for 12 months, it is basically, you can see it is 17,250, so around 17k. So this is my referral code Vani YT. So with this, you need to apply. So you can see the gift box here. So there you need to apply this code, that is Vani YT, so that you will get 10% discount on this particular offer, 17,000. So you will be getting 10% discount on that. Okay, so basically you can go for any of these months of subscription based on your convenience, how you are planning to study. So it is completely your choice. So let's get started with genetics. So genetics, let's see the different terms, like at the end of the chapter. So in Selena, okay, so they have given points to remember, so like mono, hybrid, cross, dihybrid, cross. So at the end of this particular chapter, so there are remember. So remember is given. So from there we'll discuss some of the important terms, like what is homozygous, what is heterozygous, then what is diploid, what is haploid, what is monohybrid cross, which law is applicable, what is dihybrid cross. Hi Adi, good evening. So let's have a look at the basic terms that are there. So that is from the Selena itself. So before that, let's have a look at what exactly is genetics or what is inheritance in this. So inheritance is basically passing of character, like what is being passed. So there are, the characters, like, we are resembling what is, okay, so we are resembling our parents, like, we are not completely identical, but we are similar to our parents. Okay, so in that case, so we have obtained the gene from parents, right? So in that case, so those are the character traits. So traits or characters are expressed in us because there is a transfer of allele or a gene that has taken place. Okay, and since we have got the gene from them, so basically we will have a combination of characters. So you can see here, so from father a particular gene is coming, from mother a particular gene is coming, and when both of them are together, so either this gene is going to express itself, and that is why the child is going to resemble his father. Okay, and the other case is basically the mother's gene might express. So in that case, so you can see that the child allele, yes, so this is an alternative form of a gene is called as a allele. So now if a mother's gene is expressed or a mother's allele is expressed, so in that child, so it will have a certain features from the mother. Okay, so this is basically inheritance is passing of character traits, that is from parents to progeny. So this can be allele passing itself. Okay, so that is from one generation to the next. So now this process we call as a heredity. Okay, so hereditary characters when we mention, so basically allele is transferred through the gametes mainly. Okay, and that is how the fusion is done. So inheritance is studied by the branch of genetics. So who is father of genetics? Who is called as the father of genetics? So though the progeny has major characteristics of species, so there are basically, you can find a variation. So what is that variation? Now this child is not completely, like, identical to a father, so it is not completely identical to a mother. So in that case, yes, it is Mendel, yes, Gregor Mendel. Okay, yes, so he is father of genetics. So in that case, you can see this child, we can use the term as similar because some features from the father, let's say eyes are from the father, maybe curly hair is from the mother. Okay, so like, now this child can have a black eye from the father and a curly hair from the mother. It's like it is a combination. So combination of characters you can find from both the parents, not just one parent. So that is basically depending on the gene that is getting expressed. So let's have a look at these terms. What exactly is monohybrid cross, monohybrid ratio, then dihybrid? So I have just made it simple here. Okay, so we'll discuss one of, each one of them one by one. So this is taken from the Selena textbook. Okay, so all of this is taken, at the end. So all these terms are taken. So this is taken from the Selena. So let's have a look at monohybrid cross. So which law is applicable for monohybrid cross? Anybody? So monohybrid, a cross between two pure breeding different varieties of an organism taking the alternate traits of one single character. So that means a cross is done between the pure tall plant and a cross is done between the pure dwarf plants. So this is law of inheritance. Yes, so all of this, law of dominance, law of purity of gametes, or law of segregations, what we call, or the law of independent assortment. So all these are, all of these are law of inheritance itself. for especially for monohybrid cross, two laws are applicable because we are studying only one character. What is the meaning of monohybrid? Because we are studying only one character. So this is the character now, that is the tallness, like height is the character here. So this is a trait now. So trait being tall, the trait being dwarf. Okay, so in that case, for one character always two laws are applicable from Mendel. So that is law of dominance. Okay, and other than that, so there is one more. What is that? So law of segregation, or we can also call this as a law of purity of gametes. So there is a law of purity of gametes, or segregation can also be called. So these two laws are applicable for monohybrid cross. Then what about dihybrid cross? Which law might be applicable for dihybrid cross? So the definition, so you need to know the definition: a cross between two pure breeding different varieties of an organism taking the alternative trait. Okay, so what is that? Taking the alternative trait of one single character. So this is going to take one trait from this, and it is going to take one trait from this one. Okay, so you can understand this one. The definition says here, just a second. So for dihybrid cross, it is law of independent assortment. Okay, so only this law is applicable for the dihybrid cross because two characters are there, so independently they are getting assorted. Okay, so law of independent assortment is applicable for this one. For this one, mainly law of dominance. Anyway, here also law of dominance plays a role, but two characters we are studying. So when two characters have to be studied, it is a law of independent assortment that is applicable for dihybrid. So since it is said dihybrid, that means two characters are studied at a time. So in that case, it is law of independent assortment. So I'll just explain that. Then there is a monohybrid. So only one character is studied here. So this is a law of dominance. So here you can see by the exam, like by the definition, two pure breeding different varieties, tall and dwarf is taken. Then when the progeny is produced, this is the progeny produced. Okay, so that is taking the alternative traits of one single character. So this is taking an alternative trait from one single character, like one is coming from here, the other is coming from here. So this is now all the plants which are produced are anyway tall. Okay, so let's do a Punnett square for this one. So this is TT now, and here is TT. Okay, so you can see that all of them have taken a alternate character from each of these parents. Okay, so now all of these are tall itself. So this we call as a monohybrid. So why it is monohybrid? Only one character is taken for a cross, and that character is what? The character is height. Okay, so in that the traits are what? The traits are tall trait and then the dwarf trait. Okay, so moving on to next one, monohybrid ratio. So what is yes, independent assortment, there will be, yes, that's right. Okay, so monohybrid ratio, the kind of ratio obtained in the progeny, so that is in the F2 generation by crossing two different traits of a single character. So now this is F1 generation, we can say that the genotype is for F1 generation. Okay, so genotype is what? Anyone? So what is the genotype of the F1 generation? And what is the phenotype of the F1 generation? Any idea? So genotype here, no idea? Okay. So genotype is this one. So this itself is a genotype. Okay, because this is TT, like one is capital and one is small. So this is the genotype. Okay, so ratio, I'm not asking about the ratio because here you won't get any ratio. So in F1 generation there is no ratio because you can see all of them are same. Okay, so it is 100% all of them are TT itself, and here also all of them are tall. So phenotype, you can just write it as a heterozygous tall. Okay, so this is a heterozygous tall. Why we are saying heterozygous? Because okay, so why we are saying heterozygous? Because you can see one is dominant there and the other one is recessive. Okay, so this is not a homozygous condition, so this is a heterozygous condition. So now we need to go for the F2 generation. So what will happen in F2 generation? So we are making a cross between the product that we have obtained. So in that case, a cross is made. So now you can see this is the one. So in this case, alright, so now you can see this is TT and here it is TT. So this is TT again and this is TT. So now you can see homozygous tall, heterozygous tall, and then there is homozygous dwarf. Okay. Yes, so in that case what we can say, so genotype, phenotype. So let's say they are not asking a ratio. Okay, so monohybrid ratio will tell you about a kind of a ratio obtained in the progeny. Like, you know, it will tell you like how many this type heterozygous are there and how many homozygous are there. But here in this case, so we are just trying. So first let's write a genotype and a phenotype for this. So what is the genotype of this one? Anybody? Phenotype also, ratio also I'll explain. So what is the genotype and phenotype here? So genotype will just write it as this. Okay, so TT and this is TT. So this is the genotype. Phenotype you can just write that one is homozygous tall, the other two are heterozygous tall. Okay, so this is heterozygous tall and one is homozygous dwarf. Okay, so this is the phenotype. So you are just writing it there. Okay, but when you are writing a phenotypic ratio or a genotypic ratio, so first we'll write a genotypic ratio here. Okay, so what is the ratio, genotypic ratio? So 1 is to 2 is to 1. Okay, so why? Because one is homozygous tall, two of them are heterozygous tall, and one of them is homozygous dwarf. Okay, so why we are saying homo here? Because both of them are same. Okay, so both the alleles of a gene, what are they? So they are all like, you know, dominant. So here both of them are dominant and here both of them are recessive. Okay, so why do we say heterozygous for this one? Because, you know, this is called as a heterozygous because hetero is different. So you can find a different alleles here. So one is a dominant allele and the other one is a recessive allele, and that's the reason we call this as a heterozygous. Okay, so only you can just looking at the genes you can mention that whether it is a homo or a heterozygous. Yes, so this is genetics chapter. Okay, so phenotypic ratio. Now what is the phenotypic ratio? So in this case, how many, like, you know, now if you are trying to grow this plant, let's say you are collecting these genes and, you know, you have a seeds of these plants. So when you grow them, there is a plant growth, right? So now, you know, this plant, let's imagine this as one seed now. Okay, so this as another seed now and this as the another seed. So now when you put them in soil and then when it grows, all three of them are gonna grow tall itself. So that is three of them are tall, and this seed if you are taking, okay, so let's imagine this as a seed. So now you put it in a soil. So this is, you know, even after a maximum growth is achieved, so this will still remain a short plant itself. So that is a visible character. So what is phenotype? So phenotype is basically a visible character. So when you grow a plant and when you observe it, so you can understand like this plant is short and this plant is tall. So that is phenotype. Genotype is genetic makeup. Just by observing you won't understand that. Okay, so this has to be done, like, you know, it has to be analyzed in the laboratories, like what exactly is the genotype, or just by crossing, you know, different plants. So we can understand that there is a allele for the short gene as well. So 3 of them, no, we cannot say 3 of them are dominant. Yes, so anyway, there is a dominant character, and that's the reason all of them are growing as a tall itself, which is a dominant character. But it is not completely dominant, because one is heterozygous, like heterozygous condition you can say. But we don't use the term as dominant and recessive here. So 3 of them are tall and one is dwarf, we will say. So, or if we are saying with respect to dominant and recessive, then in that case the genotypic ratio what you are mentioning. So you can see here, 1 is homozygous tall, 2 is heterozygous tall, and 1 is homozygous dwarf. So that term we can use here. Okay. So not completely dominant or a recessive. So this is the ratio, the kind of a ratio obtained in the progeny. So that is in the F2 generation by crossing two different traits of a single character. So let's have a look at the dihybrid cross. Alright, so now I told you guys that monohybrid cross, only law of dominance is applicable, and other than that, so there is law of segregation also applicable. So what is the meaning of that? So how do we say that the law of dominance is applicable, or law of law of segregation is applicable? So I'll just tell you this. So now this is the condition. So this is the progeny that we obtained in the F1 generation. Okay. So now why law of dominance is applicable for this one? Because you can see that law of dominance is what? Like out of a pair of a contrasting character. So this is a contrasting character now. Okay. So why is it a contrasting character? Because you can see one allele for tallness, the other allele for dwarf. Okay, so that is short. So in that case, now out of a pair of a contrasting character, so one is going to express itself, so that we call as a dominant allele, because this is expressing itself, and that's the reason we call this as a dominant allele. The other pair, what is this? So this is not expressed, so this is being suppressed. So in that case, this we call as a recessive allele. Okay, so why do we call this as a recessive allele? Because this is not expressed in a heterozygous condition. So out of a pair of a contrasting character, when we mention, it is basically a heterozygous condition itself, not a homozygous condition. If it is a homozygous condition, what will happen? So both of them are expressing. So this is also dominant, this is also dominant, and both of them together are expressing. So tall plant is there. But in this pair, it is a heterozygous condition. So here what happens? One is expressing itself, the other one is not expressing itself. So out of a pair of a contrasting character, the allele which is expressing itself, so that we call as a dominant, and the other one which is not expressed, so that we call as a recessive one. So what does the law of segregation says? Anybody? What does law of segregation says? Law of segregation, or it can also be called as law of purity of gametes. So you need to write the laws like as it is given in your textbook. Okay, so that is like it is mentioned. Law, you cannot write your own sentences for a law. Okay, so in that case, whatever is mentioned in your Selena textbook, so you can go according to that, the one which is mentioned there. Okay, so always a segregation occurs, like before the gamete formation. So I'll just explain it here itself. Okay, so now this is a condition. Okay, so in this case, both of them are not coming together. Okay, so there is a segregation. Okay, so this is getting separated. So now this is one allele. So let's imagine now, just a second. So I'll just explain it in the form of a chromosome. So now these are a homologous chromosome, or this is the, okay, one second. So there is a chromosome now. So in this one, you can see that this particular chromosome, what is that? So out of this chromosome, so one is having T, the other one is having T. Okay, so this is what now? This is a diploid condition. That means two sets of chromosomes are there. Okay, character, gene and allele and trait are same? No. No, they are not same. I'll just explain you that, just a second. Okay, so gene is there. Okay, so this gene is responsible for expression of a character. Okay, so character is expressed by, like, a particular gene will be there. So in that you can find different forms, like, alternative forms are alleles. Okay, so these alleles are the ones which are going to express for the different forms of a character. So that is a trait. Okay, so in that case, now let's take an example. So that is character here is height. Okay, so example, let's take the character here as a height. So now this gene which is there, so this is responsible for the height. Like, one goes, one grows completely tall and one is completely short. Okay, so there is a gene, and in that gene there are two forms. Okay, so you will find two forms. So the alternative forms are called as a allele. Okay, so in that two forms, what are there? So one is, like, one is completely growing tall, the trait, the tall trait, and the other one is a dwarf or a short. Okay, so in that case, now if this allele is expressed, then in that case this individual is going to be too tall. Okay, if this allele is expressed in that gene. Okay, so if this allele expresses itself, so in that case the other individual who's there, so he is going to be short. So now depending on which type of a gene is expressing itself, you can find individuals with the different height. So that is tall height, or short one. So that is completely depending on what type of a allele. So now this is a gene. Okay, so in this gene a particular sequence is there. Okay, so this is the alternative form of, this is one sequence and this is one sequence. So this we can call as a allele. Okay, so this is a allele in this gene. Okay, so this is from two different chromosomes. So now this is one chromosome and this is one chromosome. If this is expressing itself, so in that case tall trait you can see. If this is expressing itself, you can see a short individual. So this is about the alternative forms which are expressing itself and the alternative forms of a character which are visible. Is that clear, Minu? So law of segregation I was explaining, just a second. Yeah, so law of segregation is basically a sorting out, like, now out of a pair of contrasting character, both of them are not going together. Like, if we have to make a cross here as well, like, we need to make a cross between this one. So you can see that these are getting segregated, so they are getting sorted out. So here also you can see this is separate, this is separate. So now we have sorted them out. So in that case TT is there and it is crossed with TT here. So these will get sorted here. So this is a segregation, and here also this is getting segregated. Then only you can see a combination, because this is a 2n. Okay, so that is two sets of chromosome, which is a diploid condition. Here we don't want two sets of condition, because it is for any gametic, or we can say any body character expression, only one allele is going to express. Not both of them are anyway might be expressing, so that is only in a homozygous condition. But if it is a heterozygous condition, always they'll sort out first, like, there is a segregation that can be seen, and then they'll be expressing themselves individually. So before the expression occurs, or maybe when we are saying that the gamete formation is occurring, so these two will always get separated. Okay, so they will get separated, and then only we can find a different combination. So we can find, as they are separating, so we can find this combination. So if this and this combine, so this is the combination. If this and this combine, this is the combination. And if this then this combine, this is the combination. And that is how all the children of a parent are actually different from each other, because there is a segregation in the genes or alleles is gonna occur. So genes or alleles, what are they? So they are gonna segregate. It is not gene actually, it is the allele. Okay, so alleles are getting segregated before the progenies are formed. So that is how you can find all the children who are produced to a different same parent itself. So they are all different. So that is mainly because the genetic recombination occurs, that is by a segregation. The next one is gene. So I hope you understood about the gene. Yes, so gene की वजह से character होता है and allele की वजह से trait होता है. Yes, that's right. Okay, so now gene is basically the basic unit of heredity itself. Okay, so it is a specific part, so that is the DNA segment. So you have studied about the DNA, right? So DNA being a coil structure. Okay, so in this DNA, a part of it, so now this is a part, just a second. Yeah, so now let's say this is the part. Okay, so this is a gene now, a part of a DNA, a basic unit of a heredity, or we can say it is a specific part of a DNA segment. So that is in a chromosome. So now this is one part, like it will be paired here as such. So that means this whole part. Okay, so now this is a gene which is expressing a eye color in your body, like so some will have brown colored eye, so some can have black colored eye. So that is because depending on a type of a allele that is expressing. So in that case you can see that one can have a brown eye or maybe a black eye. So this is a gene. Okay, so gene is basically a short segment of a DNA itself in a chromosome which is expressing itself. Now allele, is this clear? Any doubt till here? Any of you? So I'll just erase this, just a second. Yeah. So allele you can find. So allele is what? It is the alternative form of a gene. So let's see here itself. So I'll just explain it to you here. So now there are pair of a chromosome. Okay, so this is a chromosome now, and there is another chromosome. So this is, let's say this is a homologous chromosome. Okay, so what is homologous chromosome? Anybody has any idea what is homologous chromosome? So this is a homologous chromosome. So when do we say that this is a homologous chromosome? So that we say they are homologous when both of the chromosomes are of same shape and size. Okay, so this we call as a homologous chromosome. So now there is a alternative form from this gene. Okay, so this is one allele now, and this is one allele which is responsible for expression of what? Expression of a eye color. Okay, so now there is a gene. So this is a gene on the whole. A part of a gene is actually a allele. Okay, so here also a gene is there. A part of that gene is a allele. So alternative form. So one allele is responsible for I'll just write it here. So this allele is responsible for brown eye. Okay, and this allele is responsible for black eye. Okay, so I'll just erase this one. Yeah, so now this allele is responsible for black eye. Okay, so in that case you can see that since this is going to dominate, so you can find an individual. So now these are the two chromosomes that is found in an individual's body, but this is going to express itself. So this is the alternative form of a gene. So this is one allele, this is one allele. Okay, where you can see the gene is mainly for the eye color, and the alleles are the ones which are expressing the trait. So trait is what? There is a black trait and there is a brown trait. Okay, so this will be dominating itself, and based on that we can see that the individual is having a black eye. Okay, so this individual will have a Black eye because this allele is expressing itself. So the trait that we see is a black eye, which is a dominant allele. Okay, and now in the other case, this allele is not expressed in itself. So this is being suppressed, or this is a recessive allele. So in that case, you cannot see an individual having a brown eye. So that is because in his body, even though both the alleles are there, the allele for the black is also there, allele for the brown is also there, but you can see his eye being only black but not brown. So that is because it is expressing in his body, and that is why we call it as a dominant. And the other one is not expressed, that is brown one is not expressed, so that we call as a recessive allele. Is this clear about the allele? Any doubt? Minu or Adi, any of you who are here? So this is about the gene or allele. So free and attached earlobes of the earlobe character. So that is, tongue rolling can also be considered in this. So dominant allele and recessive allele I already explained. Okay. So dominant allele is super ruling allele of the gene. So this definition you can write. Okay. So what is that super ruling allele of a gene? Because this is expressing, like, in your home, so you have a younger sister and you are the one, so you are the elder one, like, usually parents are going to put the younger one because she or he is the small one. So in that case they are ruling, like, their things are happening but not your things. So that is because they are dominating you. Okay, so in that case one allele is going to dominate, the other one is suppressed. Okay, so that suppressive allele is called as a recessive allele, the one which is dominating, so that we call as a dominant allele. So super ruling allele of a gene, so allele for tallness of a pea plant, because this is expressing itself. Okay, and here the suppressed allele of a gene, so that is, it is not expressed, so allele for the dwarfness of the pea plant. Then you might think that, okay, so you might think that, so if always the tall one is expressing itself, then why do we have a dwarf plant? Yes, so we have a short plant also, no? So we know that the tall is always expressing itself because that is a dominant one. Then how come we have a dwarf plant? So that is because the tallness will be expressed in this condition. Okay, so that is a homozygous condition. Now in that case both of the alleles are for tallness. Okay, so in homozygous condition, though, all the plants which are going to be produced, they'll be tall itself. Now we'll take this condition. So this is a heterozygous. So this is what condition? This is homozygous condition. This is heterozygous condition. So here you can see that one is for tallness, the other one is for dwarfness. So in this case this is expressed and this is not expressed. So since this is expressed, so this particular plant is also going to be tall itself. Then how do we have a dwarf plant then? So dwarf is expressed only when both the alleles are dwarf. Okay, so this allele is also dwarf and this allele is also dwarf. Only then you can see that the plant being dwarf or any individual being dwarf. So that is mainly because both of the alleles should be there and both of them should be expressing themselves. So only in that case you can see that the recessive character is expressed. Okay, so now we can just write it here. So that's partition this. Okay, so recessive character expression you can see only when this condition can be seen. So that is the dwarf one. So these are the terms which are mentioned. Just a second, I'll drink some water. Is it clear? Any doubt? Okay. So let's see this. Okay, so now you can see there is a okay, so dominant allele we discussed and recessive allele we discussed. So next term is homozygous. So this I already told you, like what exactly is homozygous. A condition in which a pair of chromosome carry similar alleles. Okay, so there are similar alleles of a particular character. So what is that similar allele? So like both of them should be capital T, or both of them should be small t's. Okay, so that is a homozygous condition. Okay, so here they have given an example. A colorblind daughter will have both the X chromosomes with the defective allele, then only female will get a disorder. Or otherwise, female getting a disorder only with one faulty chromosome, so that is not possible. So if both of them are expressing themselves, so then only a disease can be seen. Okay, so let's see this. So now a homozygous. So what exactly is homozygous condition? So let me just explain you this. So what all different examples can be taken for this one. So homozygous is this one. So let's write what is homozygous, like all the examples which are mentioned in your book. So I'll just write them. Okay, so this is a homozygous. Okay, and this is a homozygous. Okay, so this one is a homozygous. Okay, and we can say this one is a homozygous. All right, then what else is given? Rolling. Okay, so there is tongue rolling. So in that case, this is a homozygous, this is a homozygous. Okay, so that means what? You can see that both the alleles are same. Okay, so similar alleles are there. Okay, so you can see the definition here. A condition in which a pair of chromosome carry a similar alleles of a particular character. Okay, so similar alleles you can find in there, so that we call as a homozygous condition. So what else example is given in your textbook other than tall, dwarf? Then XX is there, then R, tongue rolling capacity is there. So maybe we can take some more, like this is for tall character. Okay, so this is with respect to height, this is with respect to we can say maybe X-linked inheritance, we can say. All right, then other than that, this is for tongue rolling. Okay, so now depending on, so you can see that the dwarf plant is expressing itself because both the alleles are short here. Okay, and here also tongue rolling is not possible, only when both of them are recessive ones. Okay, so this is a roller and this is a non-roller. So let's write this here. So this is a roller and this one is a non-roller. But what is it? It is anyway a homozygous condition itself. So then what is heterozygous? So when do we say that this is heterozygous? Okay, so we'll call this as a heterozygous. So that is when TT is found. Okay, or RR is found, or this also is a heterozygous condition. Okay, because alleles are different there, so this also can be called as a heterozygous. So in that case, so not like dissimilar alleles you can find in here. Okay, so this is with respect to dissimilar alleles formation, and that is the reason we call this as a heterozygous condition. Okay, so why it is heterozygous? Because one is dominant, okay, and the other one is recessive. All right, so that means they are not similar, so that is a heterozygous. So when do we say homozygous? Because when both of them are same alleles or similar alleles, so this is similar, this is similar, this is similar, this is similar, you can see this one, so all these are similar. So when similar are found, so that is a homozygous. So let's see the definition of the condition in which, hi, good afternoon. So a condition in which a pair of chromosome carry similar alleles of a particular character. So you can see that the color blindness is mentioned. Then heterozygous is what? A condition in which a pair of chromosome carry dissimilar alleles for a particular character. So now this could be the one this could be the one, okay, or we can say that this could be the one. So all these are heterozygous condition where you can find one being dominant and the other being recessive, okay. So this one is not dominant and recessive here. So this is dominant in its own way, this is dominant in its own way, because they are completely different chromosomes. If two of the X chromosomes were there like this, okay, so in that case, maybe depending on a type of allele that might be present, so we would say that this is dominant and this is recessive. But here it is not possible. This is expressing in its own way, this is expressing in its own way, okay. So next there is genotype, phenotype, okay. So what is that genotype now? Any of you? Just a second. Okay, so let's see this part. All right. So there is a genotype and phenotype that is mentioned. So genetic makeup of the plant, okay. So that we call as a genotype. So now what is genotype here? Just a second. Yep. Okay, so genetic makeup, like this is the combination that we might be having. So RRYY has to be crossed with RRYY, okay. So that is round and yellow seed, wrinkled green seed, okay. So in that case, how are we going to make a combination? Because both of these are same, so we need to take only one gamete. So that is gonna be R, and here it is capital Y. So again here both of them are same, so this is going to be R, and both of them are same, so we'll be taking 1. So that is the first generation, okay. So F1 generation product or the progeny that we'll be getting is this one, RRYY. Like since both of them are same here, so we need to form a gamete. So gamete is what? It is a haploid condition. This is what? This is diploid condition, because two sets you will find, okay. So two sets of chromosomes are there. We don't want two sets of chromosomes because we are forming a gamete now. So for gamete formation, you will be taking only one set. So that is a haploid, this is a haploid, this is a haploid, and this is a haploid. So first generation, you will find this as the progeny, okay. So this is all of the plants being round, because you can see one character being dominant here and the other one being recessive. So in that case, this is anyway round itself, not wrinkled, because dominant character is there, and this being yellow itself, not a green one, because green one is a recessive trait, yellow one is a dominant trait. So all of them are round and yellow. Now to make a cross in the F2 generation, so we need to form the gametes again from this one, okay. So how are we going to form a gamete here? So let me just explain it here, okay. So now this is the one that we have got. So this is the product that we have obtained. So we need to form the gametes now, okay. Hello, okay. Alright, so let's have a look at this. So now this is the combination. So we need to form a gamete so that we can make a cross for the next generation, that is for the F2 generation. So how are we going to make a gamete? So now we need to take this R and we need to cross it with one Y, okay. So because we will be considering only one. So what will form now? RY, okay, because this is the capital Y and this one being the small y, okay. So now we have not finished yet with the R. So there is one more here, because this one will take the same R and the other Y. So in that case you can see that R, one second, yeah. So R and Y, okay. And the other one. So we are done with this R now. So next we'll go to the other one. So this is the other R. So in this case you can see that the small r we are combining it with the capital one. So this is RY, and the other one here. So this is R and Y. Okay, so now we have these gametes. So now by these gametes we need to make a repetition. Okay, thank you. Okay, alright, so you can see this one RY, RY, RY and RY. So if you don't remember, so there is one more trick that you can use. Okay, so let's say you don't remember, but anyway this is the right method to do. Okay, or otherwise let's say you don't remember like how to go, like how to form a gamete. So you can just write it as two capital R's and two small r's. Okay, one capital Y, one small y, one capital Y, one small y. So these are also the gametes. So same gamete you will write on the other side. Okay, so this is small y now, the same way the capital Y, and here it is the small y. So this way also you can make a combination. So I'll just write it neatly here. So two of the capital R's and two of the small r's. Okay, one capital Y, one small y, one capital Y, one small y. Okay, so if you don't remember like how to form a gamete, okay, but that is the way, like this is the way you form a gamete. If you don't remember, like if you are getting confused, you can just follow this. Okay, so in that case again the same thing can be repeated. So R2 capital R, 2 small r. So one capital Y, one small y, one capital Y, one small y. So you can go with this, and then you can just put up a Punnett square and write the combinations here. So that will give you a different progenies, where you can see a phenotypic ratio being 9:3:3:1. So genotypic ratio is not given, which is actually a complex one. If they won't ask, like they won't ask what is the genotypic ratio. But if they are asking, okay, so usually it is not asked, so you can just write it as a complex. Or otherwise let's say they would ask it, so it is just basically 1:2:1, 2:4:2:1, 1:2:1. Okay, so this would be the genotypic ratio for the F2 generation. Okay, so this is genotypic ratio, which they won't ask anyway. Okay, but then anyway phenotypic ratio they'll ask. So this is the phenotypic ratio. So after you make a combination of these genes, so you will get that phenotypic ratio there. All right, so let's have a look at this. So genotype and phenotype we discussed. So what exactly is genotype? So genotype is basically a genetic makeup of a particular plant or any organism. Okay, so combination of gene in an organism or a genetic makeup is called as a genotype. Phenotype is a visible character, like you can see when a plant is grown, so you can understand that this is a tall plant and this is a dwarf plant. So that is a visible character, or we can say expressed character, which is genetically controlled. All right, so moving on to variation. So what exactly is variation? मेरा क्वेश्चन है कि रिसेसिव है RY, मे बी R रिसेसिव होगा. So this is recessive, like in a R and R when we mention. So this is the dominant one, this is the recessive one. So this is dominant, okay, and this one is recessive. Yes, it is clear, Anand. Okay, so let's go with the variation now. Okay, so in case of variation, small differences between individuals due to inheritance. So what is that? Because one character, like one allele is expressed, like a father's gene is coming and a mother's gene is coming. Okay, so let's say these are the two genes, one is father's gene and one is mother's gene. So in that case, if a father's gene is expressing, so in that case we can see that the child having one of the feature from the father. In another character, let's say mother's gene is expressing, so in that case you can see that this gene is expressing, so that means the child will have a character from the mother. So is that did you get this, Anand? So I just replied you. RR recessive है. Okay, so you got this, no? So this one is RR. So this is a recessive by itself. Okay, so RR is recessive. ry में भी r recessive होगा? No. Okay, so this one I'll explain it here. So this one is dominant. So this is dominant and this is dominant. Okay. So here both of these are there. So this is recessive. Okay. And this one is recessive. Alright. So now in this case you can see that this is dominant and this is recessive character. Okay. So this one is a recessive character. That means it is not expressed. So now if you are asking about R and Y. Okay. 1 second. So I'll just explain you this. So R and Y you were asking. So in that case this is dominant. But anyway you can find that this is a completely different character. So this can express itself or depending on what type of a condition. So anyway we do not find. So this is just a gamete. Okay. So for any expression, so there has to be a combination like R Y Y condition is there. So in this case now one is dominant, the other one is recessive. Okay. So here also one is dominant and the other one is recessive. So dominant one is going to express itself. Okay. Recessive one is not expressed. So this is not expressed. Alright, so moving on to the next one. So that is mutation. So variation is basically, so you will find different characters. Okay, so that is character differences with respect to both the parents. So in that case you can see that one of the parental character you can find. So that is with respect to, just a second. Okay. Alright, so here in this case, so you can see that the small differences between individuals, so these are due to inheritance. So in that case, so you can see that the parental, like father's gene is expressing, mother's gene is expressing, and that's the reason the child can have both the features. Like you might have seen people comparing a father, like child to a father also, to a mother also. So in that case one of the gene is expressing from the father, one of the gene is expressing from the mother in this child, and that's the reason you can find a variation. So now this child who is born to a particular parent, okay, so it is not completely similar. So it is it is not completely identical, but it is similar. So that is because both of the genes are being expressed in this case. All right. So now mutation, when we mention, so sudden change in one or more genes or in the number and structure of a chromosome in the progeny. Okay, so this is with respect to sudden change. So what is that sudden change? Alright, so what is that sudden change basically? So sudden change in one or more genes. So there can be variation. So I'll just explain it to you here. Just a second. Okay, so let's have a look at this. So now a genetic change can be there. So what is that genetic change? So basically a sequence is there in a DNA. So let's say this is a DNA. So in this DNA you can find ATGC. So this is the sequence which is there. Okay, now instead of T, okay, so instead of T you can find that, so T is not there, but instead of T, so there might be formation like T is replaced by a G itself. Okay, so in that case earlier this was responsible for expression of one particular character. So let's say maybe insulin formation was occurring, but now because of the sudden change, so it could be because of any radioactive radiation might be there, like UV radiation, so anything that has caused a mutation. So this T is not there, like it is a mutation. So we call this as a mutation now. So because because of this mutation, so you can see that okay, so you can see that because of this, so you can see that so insulin formation is not occurring. Okay, so there is no insulin formation, but instead, you know, this protein itself is not being formed. So that is how, you know, some of them we say that the diabetes is genetic. So like, how do we say that the diabetes is genetic? Because the same gene, that is the sudden change in gene, or we can see mutated gene, can follow, like, you know, it can go from one generation to the other generation, and that's the reason, you know, the next generation there is a chance that the insulin production might not occur. So this is because you can see that the protein production might not be occurring. So that is the sudden change. So sudden change in one or more gene in the number or structure of a chromosome as well, so which normally may not have existed in the parents, so also the grandparents or even great grandparents, like, you know, suddenly there can be a arrival of a new gene, that is because of a mutation, and it can lead to disease condition. So like, you know, you might find in certain cases the grandparents did not have a disease, or the parents did not have a disease, but suddenly this generation is getting a disease. So that is because of a mutation. There is no other reason other than that. So there is a sudden change that has occurred in a DNA, and that's the reason from this generation this gene will be carried, and maybe the children, the grandchildren can have a disorder. So this is because of the mutation which has occurred only in this particular generation. Okay, so albinism, total loss of a skin pigment, which can be a example. Homologous chromosome, so this I already told. A pair of corresponding chromosomes will be there, which are of same size and shape, and they are coming from each parent. So one is coming from the sperm, the other one is coming from the egg. So that is how we will have in a human. So it is 46. How 46? 23 chromosomes are coming from egg, 23 chromosomes are coming from sperm, and that is how 46 chromosomes can be found. Any doubt? Is this all clear? So before I end, so let me just say about this. So there is a gladiators batch, which is an all subject batch for class 10, and this started on May 4th. So you have an option to watch the recording, yet many more sessions which are yet to go live, so you can attend the live session. So if you are subscribing for class 10, so you will be getting class 11 and 12 subscription absolutely free. And the mode of delivery in this batch is English. Here you can see that the mode of delivery is English. Okay, so here also you can see that this started on May 11th, but yes, you have an option to watch the recording, and many more sessions which are, which are yet to go live, so you can attend the live sessions there. And this is a IPL where we'll be discussing 3 marks and 5 marks questions in detail. Okay, so any doubt you have, you can also get your doubts cleared. So this is basically, we'll be covering the important topics in the form of questions. So all these subjects will be covered. So you can, all you need to do is download the Unacademy Learner app and start taking up the classes. So never miss on any update from your favorite educator. So that is in a community. Get session note and practice material. Share your feedback directly with the educator via poll. So opportunity for all learners to report any inappropriate content in the video. Be the first one to report a particular issue to claim your prize. Report any inappropriate content using the form in the description. Okay, so you can download the Unacademy Learner app and select your class. That is class 6 to 12 will be available. Okay, so then you can include your phone number, email ID and all. So you can log in. So select your preferred language. So you'll be done. Okay, so then you can start attending the free live sessions, and those sessions which are actually there are certain courses which are not free, so those are plus subscription classes, so you need to take a subscription for that. And for subscription you can use the code VANIYT to get 10% discount. So the code is VANIYT. Okay, so with this code you will be getting 10% discount on the subscription. Okay, so thank you all. See you all in the next session. So subscribe to our channel, that is Unacademy ICSE and ISC boards. Click on bell icon to get notified about this session. Like and share the video and download the Unacademy Learner app. So see you all in the next session. Bye bye everyone.